Hello, I'm Toby. I head up product development at Nuclera in the UK. Today, I'd like to talk to you about our GPCR and new Nanodisc screening product that we're releasing. I'm going to be talking about this for the next 30 minutes or so, and then we can have a question and answer session at the end. Hope you enjoy it.
Thank you for the introduction. The title of my talk today is Assay-Ready GPCRs in 48 Hours, Unveiling eProtein Discovery GPCR Nanodiscs. Here's a quick overview of what we'll cover today. First, we'll look at the challenges in GPCR discovery and how cell-free protein synthesis is a gateway to unlocking high-quality protein. Then I'll introduce the eProtein discovery workflow, which enables you to multiplex, screen GPCR constructs and nanodiscs in parallel. After that, the main event, the exciting new product announcement of our GPCR-specific nanodisc kit. And finally, we'll look at some comprehensive case studies, including data on the beta-1 adrenergic receptor and a preview of FFAR1. GPCRs are incredibly high-value targets. Over 33% of FDA-approved drugs target GPCRs, spanning cardiovascular, neurological, and metabolic diseases. A prime example are the GLP-1 peptide agonist drugs like Ozempic and Mounjaro, which are driving multi-million-dollar weight loss treatment regimens as we speak. However, there is a massive protein gap. Out of the 250,000 structures in the protein data bank, only about 250 of them are unique GPCRs, which is less than 1%, a big mismatch compared to the number of drugs that are targeted at this important class of proteins. As a guest speaker noted in a recent webinar, structural biology tools are advancing rapidly, but our ability to produce complex proteins has not kept pace. The root cause of this bottleneck is the inability to generate purified, active and stable receptors. Simply put, you cannot characterize what you cannot produce. No protein, no structure. The first major hurdle is simply getting your hands on the protein. In a standard workflow, you start by cloning plasmids, then select a host like baculovirus, insect cells, mammalian cells, or E. coli, and then after expression, if you're able to get them to express, you move to detergent solubilization from the membranes. This step is notoriously harsh because you're ripping the protein from its native environment, stripping away lipids critical for stability and function. If you make it through affinity and size exclusion purification, you might be able to get your hand on a few micrograms of functional GPCR for validation, if you're lucky, or if you're very patient. At any step in this process, you could hit a brick wall. No yield, severe aggregation, or a completely inactive receptor. All of these are potential tripping points that will slow down your research. Then you spend 3 to 4 weeks running harsh detergent scouting cycles, only to find the solubilized receptor is dead, forcing you back to square one, trialing different constructs, different expression conditions, etc. This iterative process compounds the timelines to functional GPCRs. In fact, in a recent poll of membrane protein scientists we carried out in our February webinar with Dr. Frank Bernard, a world-leading expert in cell-free membrane protein synthesis, yield was reported as the #1 issue, closely followed by aggregation and misfolding. Cell-free protein synthesis offers a brilliant solution to this paradigm by decoupling expression from cellular constraints. By skipping the living cell, you bypass toxicity and resource issues entirely. In a cell-free reaction, you combine a core reagent containing all of the transcription and translation machinery with your DNA template. It's highly customizable. Crucially, for membrane proteins, you can supplement the reaction with pre-assembled nanodiscs, membrane mimetics, alongside other additives like ligands, chaperones, and redox modifiers. As the GPCR is translated, it inserts directly into the nanodisc. You avoid the harsh extract and rescue cycle completely, maintaining these critical lipid interactions from the start. We call this approach stabilizing as you synthesize. There are three main approaches to cell-free membrane protein synthesis stabilization with nanodiscs. The one I just described, co-translational insertion into a pre-assembled nanodisc, is the method shown on the left. It's the most popular method and the one we prefer. The other approaches involve either co-assembly of the membrane scaffold protein and lipid upon membrane protein expression, or co-expressing both the target and the membrane scaffold protein simultaneously in the presence of lipids. They all have their pros and cons, and each of them have their own benefits. We utilize the pre-assembled nanodisc approach because it is technically superior and they do not require optimization of DNA template ratios or lipid concentrations. They're just provided to you in a tube, ready to go without having to worry about it. With co-expression, you can deplete your cell-free resources trying to make two proteins at once, lowering your target yield, which is not a great thing when the GPCR targets aren't great expressors in the first place. And with co-assembly, you might have to optimize the stoichiometry of the membrane scaffold protein and lipid, which can be a complicated thing to do. And why bother when you can just get pre-assembled sent to you in a kit. With co-translational insertion using our QC pre-assembled nanodiscs, with co-translational insertion using our QC pre-assembled nanodiscs takes the trial and error out of this process before the expression reaction even starts. It's the most widely reported and successful method used in the literature to stabilize GPCRs. But what are they? It's vital to understand that a nanodisc is not a single fixed condition. It's a highly tunable design space. As you see in the diagram, a nanodisc consists of a membrane protein scaffold belt holding together a phospholipid bilayer. You can tune this environment by altering the scaffold to change the disc diameter so you can accommodate different size receptors. You can also alter the lipid composition within the belt by changing the head group charge, alkyl chain length, and saturation. These properties directly affect membrane fluidity and thickness, which ultimately dictate the folding, stabilization, and ligand binding interaction of your specific GPCR. Therefore, empirical screening of nanodisc compositions is absolutely critical for obtaining the best fit to yield high-quality GPCRs. This is exactly where our eProtein discovery system comes in. It is your friend. Our platform enables automated, multiplex screening of different GPCR constructs against multiple nanodisc compositions in parallel. On the right side of this slide, you can see this in action. This is our eProtein discovery digital microfluidic cartridge carrying out nanoliter expression and purification reactions in a miniaturized lab on a chip. It generates decision-grade data overnight that you can then scale up to large protein quantities, helping you move more quickly from uncertainty to an informed next step. Excuse me. For challenging membrane proteins, such as GPCRs, that parallelism is important because it converts slow, iterative trial and error into rapid, screen-driven workflows. eProtein discovery is built on four technology pillars. The first is cell-free protein synthesis, which bypasses cell-based constraints and enables rapid, customizable protein production. The second is digital microfluidics, which powers automated, hands-free multiplex screening across constructs, tags, and conditions. The third is an on-cartridge fluorescence assay that is designed to quantify full-length, stable protein and reduce the number of misleading false positives. And the fourth is a cloud-based software suite that gives decision-grade reporting and allows gives user actionable insight into expression, solubility, and purification performance and scale-up potential. Together, these pillars create a workflow that is fast, automated, and data-rich. This slide summarizes the overall path. First, you design your sequences in our cloud software and order the DNA constructs. Then you express and purify them in a multiplex screen within 24 hours. Finally, you use the data to scale the best conditions to microgram or milligram quantities overnight. From sequence to assay-ready protein in 48 hours. Wow. Remarkable. The key idea is that design, screening, and scale-up are connected in one coherent workflow rather than being separate, disconnected activities. That makes it much easier to move from DNA sequence to assay-ready protein in a predictable, repeatable, and convenient way. It's also worth noting that we offer both soluble and membrane protein workflows. But for this talk, we'll just focus on the membrane protein workflow, and specifically the GPCR class of membrane proteins. Let's go into each step of the workflow in more detail. The workflow starts with DNA design. You can explore up to 11 circular or linear e-gene constructs per screen. An e-gene construct consists of your GPCR gene of interest flanked by the regulatory elements required for cell-free expression on the N-terminus and detection and purification tags on the C-terminus. 3C and TEV cleavage sites are also encoded to allow tag removal if required. For GPCRs, this is especially useful because construct design often matters a great deal. You may want to compare full length with truncated variants, introduce known thermostabilizing mutations, test different orthologs, or evaluate fusion strategies such as Brill or other stabilizing partners. These are some of the strategies that we've used at Nuclera to enhance the yield of monomeric active fraction GPCRs. Once your 11 DNA constructs are ready, you prepare your 8 expression conditions that we call cell-free blends. A blend consists of the cell-free core extract mixed with your chosen membrane stabilizing additive. In this case, pre-assembled nanodiscs. You can also supplement with a second additive. For GPCRs, we often use a disulfide bond forming agent like PDI and an oxidizing condition with oxidized glutathione as a constant, ensuring those critical structural bonds are formed correctly. You can also mix in other custom additives like agonists, ligands, and even binding partners like G-proteins or arrestin, molecules that may be essential for downstream structural or mechanistic characterization. Setting up these blends takes just about 15 minutes on the benchtop. For GPCRs, one sensible strategy is to hold the redox supporting additives constant and vary the nanodisc composition, because disulfide bond formation is often important for receptor integrity, whilst the membrane environment is a major experimental variable. So this stage is where you define the membrane environments that you want to compare and create the screen that will tell you which construct and conditions are most promising for your protein of interest. With your reagents prepped, you load your 11 e-gene constructs and 8 cell-free blends onto the digital microfluidic cartridge alongside wash buffers, purification mag beads, elution buffers, and detector protein. Then the instrument takes over. Over the next 22 hours, it performs automated, miniaturized expression and purification, generating 88 distinct expression and purification data points with minimal hands-on time. We call this pipette and forget technology. Sit back, relax, and wait for the purification expression screen results to roll off eProtein discovery. This slide shows how the workflow is physically organized on the cartridge. The DNA constructs, cell-free blends, purification reagents, and detection reagents are all loaded into defined cartridge positions. The system then automates the workflow using digital microfluidics. The benefit here is standardization. Once the inputs are loaded, the instrument handles the expression and purification screen in a controlled, repeatable, and reproducible way, time after time. So you can always repeat the same experiment and get the same answer on our platform. It takes the doubt out of protein expression and purification. But the platform is not just measuring signal intensity alone for yield. It's also helping assess protein quality. Homogeneous high-intensity signal is what you want. That suggests a good expressor and a well-behaved soluble protein. Homogeneous low intensity can still indicate a well-behaved protein, just one that hasn't expressed or purified quite as well. Heterogeneous high intensity is different. That pattern suggests a misbehaving protein, often due to aggregation or precipitation. Speckled droplets like the ones shown in this slide do not purify, allowing us to use low purified yield as a direct indicator of low quality protein. So the software helps distinguish promising conditions from misleading ones automatically without you having to lift a finger. Amazing. At the end of the run, the cloud software provides intuitive data visualization reports, pinpointing exactly which construct and Nanodisc combination produce the highest yield for your GPCR, guiding your scale-up decisions. For GPCRs, we recommend scaling up a couple of different conditions for biophysical studies, such as FSEC or ligand binding. This report will help inform which combinations to choose. You can compare expression yield, purification yield, and efficiency, and scale-up predictions. The scale-up prediction plot is particularly useful as it allows you to identify how much scale-up volume is required to produce a certain amount of protein required for downstream work. Instead of relying on intuition and guesswork, you get a visual, data-driven basis for choosing what to scale up and characterize next. Armed with your decision-grade data, you can immediately scale up your optimal conditions. Simply combine the cell-free core, your winning nanodisc and additives, and your best DNA construct into a standard tube and leave it overnight in an incubator. That's it. That is protein expression the Nuclera way. The next morning, you perform a standard MAGB purification, which takes less than 2 hours, and voila, purified GPCR. In a total of 18 hours, you've successfully scaled your screen to produce assay-ready microgram to milligram quantities of GPCR protein. Amazing. If you need more yield, simply repeat or increase the volume of the scale-up reaction. The choice is yours. Which brings us up to a major product announcement. To perfectly complement this workflow, we're officially launching the new eProtein Discovery GPCR Nanodisc kit. We have expertly created 8 specific pre-assembled nanodiscs tailored for GPCRs for you to screen through to find the best fit for your specific GPCR of interest. There are 2 product formats. First, the panel screen kit. This contains the full 8-condition nanodisc panel for multiplex screening on our cartridge. The second format are scale-up nanodisc aliquots. These provide larger volume vials of higher concentrated material of selected nanodiscs. You can choose from 1 or all 8 for higher volume scale-up reactions off cartridge. They're completely compatible with our cartridge, our droplet operations, and we have a full set of kits to support them. The benefits are that they're ready to use, directly compatible with eProtein discovery, tailored to GPCR membrane requirements, and quality controlled to reduce batch-to-batch variability to improve the repeatability of your results, of your experiments. So more about the nature of the kit and what's in the tubes. We combined two proven scaffold sizes, MSP1D1 and MSP1E3D1, to accommodate diverse receptor geometries. These two scaffold proteins differ in length and hence constrain the nanodiscs to a different diameter. We pair these scaffolds with 4 highly validated lipid environments. DOPG as the proven standard for cryo-EM. You can see on the right of this image that there are the structure of 2 GPCR G-protein complexes produced using MSP1E3D1 DOPG nanodiscs. DOPG cardiolipin is the second flavor. This has been proven to enhance ligand binding in some GPCRs. The addition of cardiolipin results in more nanodisc and membrane lipid fluidity. That helps the conformational changes, which we all know are essential for active or GPCR activity. POPC and POPS act as physiological native membrane mimics. You can choose from those as well. And we also have POPC, POPS cholesterol lipid mix, which supports cholesterol-dependent GPCR function and has been proven to enrich the incorporation of a large number of complex GPCRs. We recommend screening the entire GPCR nanodisc panel to maximize your screen diversity and increase the chance of finding an optimal condition tailored to your GPCR of interest. Looking at the data, our curated GPCR nanodiscs consistently drive superior results. Here is an eProtein discovery screen across a diverse panel of 11 different GPCRs. The purple markers represent the nanodiscs from our new panel, whilst the grey markers represent alternative nanodisc compositions. As you can clearly see, our GPCR-specific nanodiscs consistently push the boundary to the right, delivering significantly higher purified yields than the generic equivalents. The important message here is that one size doesn't fit all. There is no universal nanodisc that is best for every receptor. Screening a range of nanodiscs is the easiest way to find the best fit for your GPCR. Our curated GPCR nanodisc panel provides a productive starting point for studying the expression of a range of GPCRs. This reinforces why nanodisc composition screening is central to the successful production of high-quality, active GPCRs. However, as we all know, high yield of inactive protein is not very useful. In fact, it's a complete waste of time. So when evaluating these nanodiscs, we had a look at the activity of the proteins that are expressed and inserted into these nanodiscs. We did this using a Turkey ortholog of beta-1, and we looked at both yield and functional ligand binding. Notice the grey bars. These represent other nanodiscs. A composition like DMPG produced very high yields, but when we tested its activity, the ligand binding fraction was incredibly low, around 5%. On the other hand, although using our curated compositions like POPC, POPS produced slightly lower overall yield, they dramatically recovered functional ligand binding, hitting nearly 18% active fraction, a massive improvement. We'll go into this case study in more detail soon, but it's just to emphasize that our biologically relevant nanodiscs ensure you don't just get protein, you get functional protein. To summarize this process, combining eProtein discovery with these GPCR nanodiscs gives you an automated, hands-free path to optimize GPCR protein production. By utilizing precise on-cartridge fluorescence and streamlined scale-up, you can go from DNA sequence to ready-to-use, assay-ready GPCRs for structural studies in just 48 hours. Mind-blowing. And here's a case study. Let's have a deeper dive into that Turkey beta-1 case study to see this entire workflow in action. The goal here was to move rapidly from DNA to a monomeric functional receptor preparation. Beta-1 is a useful benchmark GPCR. It's a clinically relevant receptor with clear pharmacological importance, especially because beta blockers act on this signaling axis in cardiovascular disease. It is also a well-studied receptor in structural biology, which makes it a strong test case. But as with many GPCRs, conventional expression and detergent extraction workflows can struggle to preserve the lipid environment the receptor needs, leading to variable yield and poor functional quality. So the question here was whether eProtein discovery, combined with the GPCR-specific nanodiscs, could produce highly active beta-1 more quickly and more reproducibly. For the design stage, 3 beta-1 variants were compared. These included human full-length construct, a human full-length thermostable construct with some mutations, and a Turkey truncated thermostable ortholog. This is our workhorse, our benchmarking beta-1. These constructs were screened against a set of pre-assembled nanodiscs, including conditions from the GPCR-specific panel and comparison conditions from other compositions. The point here was to elevate both construct design and membrane environment together because they can both strongly influence the outcome. The 24-hour multiplex screen clearly identified that the Turkey truncated variant delivered significantly higher purification yields across the board. Furthermore, the negatively charged lipids, DOPG and DMPG, were the top performing environments based on yield alone. Armed with this data, we moved straight to scale up so that we could do some of the activity tests. We did a small scale scale up of all of the candidates for the Turkey variant, delivering about 100 micrograms per mil purified receptor in less than 10 hours. Less than 48 hours, sorry, not 10 hours, with the DOPG nanodisc. And you can see the purified band on the gel, very high quality, high purity protein. This is what screening buys you, the confidence to invest in the scale-up reaction. Predictive yields from screen to scale, guiding you to make the protein you need for downstream assays and structural biology experiments. Because we generated excellent yields across multiple conditions, we had the option to test them for functional activity using ligand affinity chromatography. We bound the sample to an alprenolol agarose column, washed away the non-binders, and eluted the active receptors with isopranoline. The data confirmed what I showed earlier. Whilst DMPG nanodiscs gave high yields, the protein it produced had very low ligand binding activity. Yet the Nuclera GPCR specific nanodiscs, specifically the DOPG and the POPC-POPS variants, significantly enhanced GPCR activity, reaching up to 18% functional ligand binding. Not stopping there, we decided to concentrate the protein, and these proteins are very stable when embedded in nanodiscs. We can concentrate them to greater than 1 mg per mil. We did that, and we froze grids and captured a cryo-EM QC data set. We took about 300 movies and counted about 90,000 total particles, 44,000 of which were occupied with GPCR embedded into nanodiscs, which was amazing. And 50% occupancy is quite remarkable. So super happy with that. And this demonstrates a rapid workflow path to high quality cryo-EM grids without the requirement or need for harsh detergents, which can damage the protein and impact the ability to get the structure. We've actually generated a slide on how to, we've actually generated a guide on how to get from eProtein discovery screen to excellent cryo-EM grid samples for structural biology that we are happy to share. Reach out to us in the chat if you want us to send you a copy. Happy to do that. For GPCRs, early prep can contain a mix of active and inactive populations. FSEC is a great way of confirming the percentage of active monomeric GPCR you have. Typically, you'll see 3 peaks in the SEC chromatogram. The first peak comes out in the void and corresponds to aggregated protein and nanodiscs. The second peak we've attributed to misfolded GPCRs or GPCRs that are misinserted into nanodiscs. And the third peak on the right-hand side, the lightest of the 3 peaks, that is the active monomeric fraction. So that's what we're aiming for. We'd love all of our GPCRs to be 100% peak 3. In reality, that's very, very difficult to achieve. But it's a challenge that we are accepting, and more news about that later. Knowing this information, we then perform the standard SEC polishing step to enrich the monomeric functional receptor. Here, the SEC separated aggregates for monomer and increased the functional fraction 3-fold, from 15% to 45%. The practical takeaway here is that the workflow gets you to a strong starting sample quickly, and then a simple polishing step can substantially enrich the active monomeric fraction that you need for high-quality cryo-EM structural work. So, what is the overall advantage of our GPCR expression screening technology? The main benefit is compression of the time it takes to develop that GPCR purification process. Instead of spending weeks on host optimization, extraction, and serial membrane condition testing, this workflow compresses those cycles into a handful of days. It delivers assay-ready protein in about 48 hours. And in the beta 1 example, it enabled, screened, scaled, and functionally enriched a receptor in under 72 hours. Amazing. That means faster decisions, better starting material, and a more efficient route to both functional studies and cryo-EM structural work. And remember, this was all enabled through the combination of eProtein discovery workflow paired with our new eProtein discovery GPCR-specific nanodisc product that's available to buy now. Before we open it up to questions, I want to leave you with a quick preview of another case study that we've been working on with some collaborators at Diamond Light Source, Andrew Quigley and Harish. This is about the free fatty acid receptor FFAR1. Using our system, they produced approximately 100 micrograms per mil of high quality FFAR1 at over 90% purity in just 48 hours. Using ligand binding assessment with NanoDSF, they confirmed correct folding and activity, showing clear melting temperature fluctuations that you would expect to see upon the binding of ligands such as TUG424 and TAC875. Furthermore, negative stain EM revealed excellent sample homogeneity. We'll be releasing the full application note very soon with more data included, so stay tuned. Fingers crossed, but we're hoping to reconstruct a high resolution cryo-EM structure of this APO enzyme. Really exciting. And that's it. Thank you for listening. I hope this has been informative and has given you an insight into some of the new stuff that we're releasing at Nuclera and how our technology can revolutionize the way you work with membrane proteins and specifically GPCRs. Thank you very much. I'll be happy to answer any questions you might have now.